Well, welcome back. We've been learning about life functions and we've come to reproduction and growth. And so when you look at how living things develop, when you think of yourself and your own history over the course of your lifetime, you're not the same size that you are now than when you were 2. And we call that growth. Well, how does that occur? Basically 2 things. And at the level of the cell, you see both of them. Cells get larger, so we get larger. Also, cells are going to reproduce. So there's going to be many, many more in your lifetime as you're an adult compared to when you were 2 or 3. Cells divide, so there's more cells. Cells can't get really, really large because of the limits that the cell membrane places upon each cell. Things can only come across the cell membrane so fast, and they can only diffuse into the center of the cell so fast. Things like oxygen and glucose, things that all of us need for life processes. And so cells are going to be limited in terms of their size. And so the only way that organisms can grow large and have many different types of tissues is if they reproduce. So cells divide during a process known as cytokinesis. When we look at this word mitosis, we're really talking about the division of the nucleus. And so specifically, mitosis is only going to occur in eukaryotic organisms. So that would be me, you, plants, and yeasts, all other animals, and so forth. Okay? Cytokinesis is really the division of the cell, including the cell membrane and the cytoplasm. But of course, mitosis, the division of the nucleus, must occur prior to cytokinesis because every cell is going to need its own nucleus, at least one. Sometimes you have more. And that's what you see pictured here. Well, how does that occur? And as you can imagine, it all gets down to the chromosomes. Now, you may have remembered that when we are looking at DNA, nucleic acids, and so forth, you may remember this double helix structure that you see pictured here, over on the left of the screen, right above me. And so these molecules do not get tangled up in the midst of each other. During this process of mitosis, in the preparation of that, these are not only going to be replicated during the S phase of interphase, but they're going to be organized in a very specific way. They're going to wrap around these proteins, these purple spheres here known as histones. And then that whole structure is going to coil up into a molecule known as chromatin. So chromatin is really a combination of the DNA double helix coiled up around these proteins. And so it's done in a very organized way. And so when we see this process of mitosis beginning to occur under the microscope, we can actually see these structures condense, in other words, shrink down, so they become organized. So the process of dividing up the DNA and then the nucleus becomes pretty straightforward for the cells. They can do this fairly rapidly. So DNA plus proteins is what we'll refer to as chromatin. Okay, and one half of the duplicated chromosome is referred to as a chromatid. Keep in mind that during the S phase of interphase, the DNA is going to make a copy of itself, and that becomes organized into 2 sister chromatids that are bound to one another in a structure known as the centromere. Of course, biologists, we have words for everything, but that's the important thing. Okay, and so this is a typical chromosome that you see structured here. Sister chromatids, one of these is going to be the mirror image of the other. Okay, so that's what it's going to look like. And we can see these under the microscope in a not too high magnification. It's not hard to see. And so we're looking at 2 genetically identical cells being produced from these processes of mitosis and cytokinesis. So basically all the cells in your body and mine are identical to all the other cells that we find in our bodies. Okay, yours is different from mine, of course. And so interphase, that large stage that includes gap 1, and then synthesis and gap 2, we group those together to call that interphase. And that's where the preparation occurs. DNA is duplicated. Typically, that's what you're going to see here. And so, as you recall, human beings, they have 46 chromosomes typically. Well, after synthesis, which is a later stage of interphase, we actually have 92 chromosomes because they're going to be divided up in this process of mitosis so that the resulting daughter cells each have 46 chromosomes, and we're back to the number that we started with. So this is a typical parent cell that's about to undergo mitosis. The chromosomes have already divided, and then these structures known as centrioles are going to appear, and they're going to help organize this process forward. And so mitosis then becomes divided into these 4 phases that I'm going to walk you through right now. All right, the first of which is prophase. And there's basically 3 critical events that we observe during prophase, okay? And they're very early. First, it's the appearance of these centrioles, and these lines here, these projections, are protein fibers that are referred to as spindle fibers, okay? Just like the structures on a spider's web. And they're going to begin to appear, and we're going to see the role that they play in this process later on. Also, the nuclear membrane is going to begin to break down and dissolve. We need to get that out of the way, or the separation of these chromosomes can never take place. And thirdly, as we mentioned before, these chromosomes, they're going to now become visible because they're shrinking together, they're condensing around those histone proteins. Okay, so that's stage 1, begins with a P. We move then on to the process known as metaphase. Again, 2 important events take place. You see them pictured here. First, the spindle fibers are going to attach to these structures that bind the 2 sister chromatids together. And then the sister chromatids, you can see, are going to line up along the middle of the cell. We sometimes refer to that as the equator of the cell. So you can see the amount of organization that must take place. And they've all lined up in such a way so that when the separation occurs next, the 2 resulting nuclei for the daughter cells are going to have a perfect copy of each chromosome. If they do not line up properly here, then those 2 daughter cells are not going to have the equal and necessary chromosomes that they need. So this is a really key stage, and if this doesn't occur properly, so either the daughter cells are going to die, or a birth defect could possibly result if this were to take place very early in the life of the individual. Sadly, that often occurs in death, but sometimes it doesn't. And then we're going to move on to the 3rd stage, which we refer to as anaphase. And this is where you actually see this separation. When you're watching this live, it's pretty amazing because this only takes a few seconds to see these chromosomes migrate toward the centrioles on the opposite sides of the cell. So this is where the separation occurs. And so if this doesn't come off perfect, then we're going to have an unequal number of chromosomes resulting in the 2 daughter cells. And then finally, the 4th and last stage of mitosis is telophase, where you have, again, several important events are taking place. Those spindle fibers are now going to release from the chromosomes, and they will degenerate. We have the forming of a new nuclear membrane on each of the daughter cells. Okay, so we now have 2 intact nuclei. And then the chromosomes themselves, they're going to begin to uncoil off of those histones. So then they could go to work in the process of these 2 cells beginning to grow. They're going to begin, very soon after this, beginning to copy the information found in the chromosomes of the DNA, and they're going to begin making proteins again. And the cell is going to begin to grow. But there's one very key event that must take place which occurs next. And you can probably anticipate it. And it's a difficult kind of name once you get used to it. And that is cytokinesis. Cytokinesis. Can you say that? Cytokinesis. Cyto, of course, refers to cell. All right. And this is going to occur, again, in all eukaryotic organisms where you have two very important events. The cytoplasm is going to be divided between these two daughter cells, and the cell membranes themselves are going to pinch off from one another. So now these two daughter cells, the result, are independent of one another. Now, in plant cells, the process is similar as well, except that it may not be as dramatic. You have a plate of cellulose that begins to form the beginnings of the cell wall that separates those two daughter cells. But the process is really the same. And it's really key that this occurs, because if we don't have a separation of those two nuclei into two cells, then we can end up with one cell having two nuclei and the other cell having none. And of course, that's not going to continue. That will die. And the cell that has more than one nuclei, that does occur. In fact, it's important that it does occur when it does. For example, your white blood cells and mine, they can have typically 4 nuclei in a white blood cell. And that's because cytokinesis doesn't occur in exactly the same way. You actually have more than one stage of mitosis go through. Those 4 phases we just mentioned, that actually will occur twice before cytokinesis occurs in the white blood cells. So you end up with, multiple nuclei in the same cell. Slime molds, we see a similar process. Well, listen, thanks for watching. Bye-bye.